Hi, this is part 2 of the cell and molecular biology revision. In this second part, we will look at topics such as DNA replication, transcription and translation, gene cloning, polymerase chain reaction, mutations and cancer, and we'll look at some questions from your practical sessions. Okay, let's first look at DNA replication, transcription and translation. Questions from this topic. Which one of the following is the matching sequence for the DNA molecule shown below? So for this question, you need to know the base pairing rules. Adenine binds to thymine, cytosine binds with guanine. So here you are shown the 5' to 3' sequence. The matching sequence will be 3' to 5'. So an A will bind with thymine, T bind with adenine, guanine will bind with cytosine. And this will be guanine, thymine, adenine, thymine, guanine, cytosine, and this is 5'. So the answer will be A. In the diagram below, the processes occurring at A, B, C are referred to as, so this is the central dogma theory, right? So in the central dogma theory, DNA can replicate by itself. This is replication. And DNA can be transcribed. This is transcription. And RNA can be translated to make proteins. So your answer will be A. An mRNA sequence is given below. So this is the mRNA sequence. How many codons are present in the mRNA sequence? So 3 bases is equals to 1 codon, right? 3 bases equals to 1 codon. So here you have 1, 2, 3, 4, 5, 6 codons in the mRNA sequence. So you were using the genetic code. So this is a genetic code. Determine the amino acid sequence of the protein formed from this mRNA sequence. So let's look at AGG, right? Now, proteins always start with a AUG. So even if you look at, even here the sequence starts with an AGG, we will ignore this and this sequence and we will start from this AUG. So if you look at it here, AUG is a start codon, codes for methionine, MET, right? Then UUA is leucine, here UUA is leucine. How about CCA? CCA is proline. And finally AGC, AGC here will be serine. Okay. How many tRNAs will be required for the synthesis of amino acid in the amino sequence in part A? Okay. So how many tRNAs do you need? Each amino acid is carried by 1 tRNA. So how many amino acids are here? 1, 2, 3, 4. So there will be 4 tRNAs required. Write the coding strand and DNA template strand sequences for which sequences which coded for this mRNA. So this mRNA, okay, the template was a DNA, right? Was a 3' DNA was the template. 3' DNA was a template. So the DNA 3' to 5' was the template for this mRNA. So if this is A, the DNA will be a thymine, right? T. G will binds to a C, right? Here will be a guanine, thymine, cytosine. A would bind to a thymine. Uracil will bind to a adenine, because we are talking about RNA and DNA. So this would be a C. Okay, here it will be A, and another A here. Uracil binds to adenine in DNA. Then adenine binds to thymine, guanine, thymine, cytosine, guanine, and this is your 5' end. So 3' to 5' end DNA. This is the template strand, right? So this is the template strand. We are answering this question here, template strand. Okay. So how about the coding strand? So the coding strand is your 5' to 3', which is complementary to the 3' to 5' DNA. So this is the template strand. Okay. So a T would bind to an A, right? Cytosine will bind to a guanine. Okay, so this will be your coding strand. Okay, and finally adenine will bind to a guanine, cytosine bind to a adenine will bind to a thymine, cytosine bind to a guanine, and guanine will bind to a cytosine, and this is the 3'. And so this will be your coding strand, like that. So you can see that the RNA, the mRNA is complementary to the template strand, and the template strand is complementary to the coding strand. A double-stranded DNA molecule is shown below. Write the sequence of the mRNA transcript. Now, so this is the coding strand, right? We all know that this is a coding strand, and this is the template or the non-coding strand. So the template strand is used as a template to make the mRNA transcript. So let's say this is the mRNA we are going to transcribe. Starts with the 5'. So if you have a C will bind with a guanine, A will bind to a uracil because there is no thymine in RNA, right? So guanine will bind to a cytosine, thymine will bind to a adenine will bind to a uracil, right? And then guanine will bind to a cytosine, like that. Sorry, yeah, and so on. Okay, and finally it will end with a GCCA, and this is the 3'. Okay, so this is the mRNA transcript. Now, if you notice this 5' to 3' sequence is similar to the 5' to 3' DNA sequence, with the exception of the thymine. Thymine is changed to a uracil. So if you look at this GTC, the thymine is changed to a uracil, so it's GUC. See that? So ATC has become AUC. All the thymines are converted to uracil in RNA. So that's why the coding strand is similar to the mRNA sequence. Determine the amino acid sequence. So determine the amino acid sequence of the polypeptide protein that will be formed from the mRNA translated, if the mRNA is translated. Okay. So now, as I mentioned, mRNA translation starts from the start codon. Start codon is the AUG. This is the start codon. So this is MET. Then the next one will be CGG. CGG is arginine, like that. Okay, arginine. Okay, and then ACC. gives you threonine. There's another AUG here. So AUG once again is methionine, MET, and then we have a AGA. Okay, and AGA is arginine. Okay, how about a GUA? Okay, a GUA gives you valine, right? Then a UAG, here UAG is a stop codon. Stop. Okay, with the stop codon, the translation stops. So here you see a methionine, arginine, threonine, methionine, arginine, valine, and then you have a stop. Stop is not a amino acid, so if you don't write stop, it doesn't matter. You will not be penalized. The diagram below shows transcription taking place in a cell. Transcription. So one of the components of transcription is your DNA template, right? Okay. So here the 5 prime to 3 prime, we have strand Y. So you're supposed to find out what is strand Y, strand Z, and what is molecule P. So the 5 prime to 3 prime strand Y is known as the coding strand of DNA, right? Coding strand. And then your 3 prime to 5 prime strand Z is your template DNA or non-coding strand. Okay, this is your coding strand. So in transcription, what is being formed? What is molecule P? This will be your hnRNA. If the nucleotide composition of the newly synthesized RNA was 20% adenine, the RNA has 32% of guanine bases, 18% of adenine bases, and the rest are uracil bases. What would be the percentage composition of the base uracil? So total should be 100%. So when you subtract the 20% adenine cytosine, 32% guanine, 18% adenine, you should get 30% uracil. Part B, the 4 bases in the non-coding strand of DNA from which this RNA was derived. So let's look at this RNA. So this RNA, let's say this RNA is 5 prime to 3 prime, right? So it has got 20% cytosine, 32% guanine, 18% adenine, and 30% uracil. This RNA is derived from the non-coding strand of DNA, right? So it is 3 prime to 5 prime, right? Okay. So base pairing rules. Cytosine will bind to guanine. So if cytosine is 20%, guanine must be 20% in the template DNA or the non-coding DNA. 32% of guanine will bind to 32% of cytosine. Adenine will bind to thymine. This is DNA, so it has got thymine. Uracil will bind to adenine. So that will be your answer for part B. 20% guanine, 32% cytosine, 18% thymine, and 30% adenine. Part C. The 4 bases in the coding strand of DNA. What is the composition of the 4 bases? So the non-coding strand is complementary to the coding strand. Okay, the coding strand starts from 5 prime to 3 prime. So 20% guanine. Guanine will bind to cytosine, so it must also be 20%. Cytosine 32%. Here guanine will be 32% because cytosine binds to guanine. Adenine binds to thymine, so this must be 18% adenine. And adenine binds to thymine, so this must be 30% thymine in the coding strand. Let's look at questions from the gene cloning topic. The diagram below shows the restriction map of a plasmid vector PHS7028. Numbers in the parentheses. Parenthesis refers to the brackets. Numbers in the brackets indicate the position of the restriction sites on the plasmid. So this plasmid is 4 kb in size, which is 4000 base pair. Okay, by now you should know that 1 kilo base is equals to 1000 base pair. So question A, how many fragments, they are asking for fragments, will be obtained if this plasmid is cut with AVA1, PST1 and BAMH1. So you can see here, if the plasmid is cut with BAMH1, like that, and then AVA1 and PST1. So you're going to have one fragment here, then you're going to have another fragment here, and then you're going to have another fragment here, and finally you have one more cut here. Sorry, yeah, you have one more cut here, so there's going to be a fragment here and then a fragment here. These will be 1, 2, 3, 4 and 5 fragments. Okay, so let me write this again. 1 kb is equals to 1000 base pair. Okay, okay, so let's look at the second question, part B. What will be the sizes of fragments? They're asking you for the sizes if the plasmid is cut with AVA1 only. Okay, so AVA1 only, how many restriction sites are there for AVA1? Only one here. So if I were to cut it like that, so this is a plasmid, I were to cut it with AVA1, so what would I get? I'll get a single fragment, only one fragment. What will be the size of this fragment? It will be the size of the plasmid. So this will be 4 kb because there's only one single cut. If the plasmid is digested or cut with AVA1 and PST1, okay, AVA1 and PST1, so we have AVA1 here and then there's a PST1 here on this side. So there's going to be two fragments, right? One fragment on this side, another fragment on this side. There's going to be two fragments. What are the sizes? So if you look at it here, 180 and this is 2450. That's one fragment. Okay, so one fragment from here, this is 180 to 2450. This is going to be one fragment, and then another fragment is going to be like that. Okay, so if you were to calculate, so if I were to calculate 2450, sorry, 2450 minus 180, I should get 2270 base pair. That's one fragment. And then as a second fragment from here to here, okay, that would be 1730 base pair. So one fragment is 2270 base pair and the other fragment is 1730 base pair. Now, how do you derive at this 1730 base pair? Okay, quite simply. Now, the whole plasmid is 4000 base pair, right? So if you were to minus off the 2270 fragment, you should get the 1730 base pair fragment. Okay. If the plasmid is cut with PSD1 and Hindi3, how many fragments do you expect? So PSD1 is here, Hindi3, there are two sides, one here and another one here. So three sides are being cut, so you'll have three fragments. Okay, so here, from here to here, what is the size? 1000 minus 180, this should give you 820 base pair, right? Then from here, 2000 to 1000, this should be a 1000 base pair fragment on 2000 minus 1000, right? And then you can just take 4000 minus 1000 minus 820 to give you the size of the remaining fragment, right? So this should be, sorry, yeah, let's put it here. This should be 2180 base pair to be the size of the final fragment. What will be the size if the plasmid, size of the fragments if the plasmid is cut with BAMH1 and AVA1? Okay. Now, BAMH1, there are three sites, 1, 2, and 3. AVA1, only one side here, right? Here. So there's going to be 4 cuts, there are going to be 4 fragments. Okay, I'm going to leave it to you to calculate on your own. Okay, based on what I've explained for the first 3 sizes of the fragments. So the answer will be 1200 base pair, 680 base pair, 1250 base pair, and finally 870 base pair. Now, one important thing to note, it is very important for you to specify the units. Without the units, the answer will be 0, will not be accepted. You'll be marked wrong without the units. So please pay attention to the units. The diagram below shows the various bands of DNA obtained after a plasmid CMB1 was digested with 3 different restriction enzymes, BAMH1, Hindi3, and PVU1. Size of each band is given on the left of the diagram. So these are the sizes of each band. So this band is 4781 base pair. X and Y refer to how the electrodes were connected during gel electrophoresis. Question A. Determine the size of plasmid PCMB1. So let's look at the first lane here. Okay, the first lane. What do we see? Okay, when the plasmid is cut with PVU1, it has 2 fragments. One fragment here, another fragment here. Which means there must have been 2 restriction sites for this restriction enzyme, right? 2 restriction sites. That is why when you cut with PVU1, you get 2 fragments. One fragment here, and then you get another fragment here. Now, if I add the sizes of these 2 fragments, okay, this fragment 1, let's say, and this fragment 2, if I would add the size of these 2 fragments, I should get the size of the whole plasmid. So what is the size of the whole plasmid? 4781 plus 896. This should be 7, 6, okay, and 5, 6, 7, 7 base pair. That is the size of the whole plasmid. Determine the number of restriction sites for BAMH1 in this plasmid. Now, let me draw the plasmid here once again. Okay, like that. So we know that P PVU1 has 2 restriction sites. That is why it has got 2 fragments in the first lane. So here we are talking about BAMH1. So let's look for the last lane. The last lane is BAMH1 and PVU1. When you add these 2 restriction enzymes to the plasmid, you get 3 fragments. Alright, you get 3 fragments. But we know from the first lane that 2 fragments belong to PVU1. So because PVU1 has got 2 restriction sites. So BAMH1 must only have 1 restriction site. That is why when you cut with these 2, you get 3 fragments. 1 fragment here, okay, and a second fragment like that, and then you get a third fragment like that. Understand? So the number of restriction sites for BAMH1 is only 1 restriction site. Okay, let's look at the part C of this question. Determine the number of restriction sites for HinD3 in this plasmid. So let me draw this plasmid again. Okay, HinD3. Once again, let's look at here HinD3 and PVU1. So we know that PVU1 has got 2 restriction sites, that we know. When I cut with HinD3 and PVU1, I also get 3 fragments. So 2 fragments are for PVU1. So that means only 1 restriction site for HinD3. That is why you get 3 fragments. Makes sense, right? Okay, part D. Why do DNA molecules move in the direction of X to Y? So the question is saying that this is the negative electrode, this is the positive electrode. Why is DNA moving from negative to positive? Why not from positive to negative? Okay, the answer is DNA is negatively charged, so it moves from the negative side, okay, to the positive side. Now, why is DNA negatively charged? If you recall, the DNA is made from nucleotides, right? So if you were to recall the structure of nucleotide, so it has a pentose sugar, it has a base, right? This base is your adenine, thymine, guanine, cytosine, and on this side, okay, on carbon 5 you have the phosphate group. The phosphate group is negatively charged. Okay, that makes the DNA negatively charged. Table below shows the properties of some restriction enzymes. So here they show you the restriction, then they tell you the name of the restriction enzyme and the target sequence. So BAMH1 will look for these 6 bases and it will cut after the first G. So the asterisk is where the enzymes cut. Which of the restriction enzymes above can be used to cut the linear double-stranded DNA sequence shown below? So first we need to see whether BAMH1 can cut this DNA. We will look for this sequence, GGA TCC. So do you see a GGS? I see a GGA TCC. So BAMH1 will be able to cut the enzyme, right? So any more GGA TCC? No. Okay. Let's look for EcoR1. Is there a GAA TTC here? Nope, I don't see a GAA TTC. So this does not cut the DNA. Then SMA1, CCC GGG. You see a CCC GGG? Nope, I don't see that. Okay. How about HinD3, AAG CTT? I see a AAG here and yes, CTT, right? So HinD3 can cut the DNA. How about GTT AAC? So I see a GTT AAC here. So HPA1 can also cut the DNA. So if you were to look at it in this direction, 5 prime to 3 prime, the same, you'll see a GTTAAC for HPA1. You also see a AAGCTT in D3, and then here you see a GGATCC for BAMH1. Okay. We are now going to look at questions pertaining to the polymerase chain reaction lecture. Which one of the following shows the sequential order of the PCR stages? There are 3 stages in PCR, right? So it starts with denaturing of the DNA into single strands. So this is denaturation. Following that, you have annealing of the primers. So let's say this is the 5 prime and 3 prime end, right, of the DNA. So the primers anneal to the DNA. So this is annealing. Annealing is followed by extension of the primers, extension. And then the cycle repeats. Extension can also be known as synthesis. Following a mix-up that occurred in a hospital, the parents of a child needed to be identified. PCR and electrophoresis were employed to determine the true parents. The electrophoresis of DNA samples from each parent yielded a result as shown below in figure 7. Okay. So in other words, you have 4 sets of parents here. Okay. The child belongs to one of these set of parents. So these are the DNA fragments from the child. So state the parents of the child. Okay, we know that a child gets 50% of the chromosomes of the DNA from the father, another 50% of the chromosomes or DNA from the mother, right? So if you were to look at these fragments, these 10 fragments, okay, they must belong to the parents. So let's look at this. Okay, so this fragment is found here as well, here and here, right? Okay. How about this fragment? This fragment is found here. Okay. Like that. Okay. So now this fragment is found here and here. This one is found here. Okay, we're looking at the sizes of the fragments. Okay. How about this one? This one is found here only and here. Possibly here. Okay. How about this one? Okay, this one is found here and then here. This one is found here in this set of parents, parent A, and also found here and found here. This one is found in this parent and parent F and parent H. This one is found in parent B, parent D, G as well, and this one is found in B, C. Okay. So now if you were to look at this, if we were to look at the parents A and B first, you see that this fragment is not found in any of the parents. So parents A and B cannot be the parents belonging to the child. Okay. So if you were to look at parents C and D, this fragment, the first fragment is found in D, second one is found in C. You'll see that all the child's fragments are found in C and D, completely found in parents C and D. See, C has got 1, 2, 3, 4, 5 fragments belonging to the child. found in child, and D also has got 1, 2, 3, 4, 5 fragments found in the child. So these must be the parents. If you were to look at the parents E and F and G and H, they don't have a lot of the fragments. For example, here this fragment is not found in parents G or H, so they cannot be the parents as well. Okay, likewise, you don't see these fragments in E and F, so they also cannot be the parents. Whom does the DNA sample of control belong to? Whose DNA do you think is this control? Now this DNA belongs to the lab technician who does the what do you call that, the autoradiograph, who runs this gel. This is to ensure that the lab technician has not contaminated the samples with his own DNA. What are the steps involved in the polymerase chain reaction, PCR? State the temperature ranges for each stage. So first you have denaturation. This is the what do you call it, separating of the double-stranded DNA into single strands at temperature range 90 to 95 degrees Celsius. Next, you have annealing of the primers, right? Primers anneal. This happens at 50 to 70 degrees Celsius. And finally, extension of the primers, and this happens at 70 to 75 degrees Celsius. Study the diagram of an agarose gel below. This is after electrophoresis, and answer the following questions. Samples A through F are DNA obtained from different individuals. So these are samples from, let's say, 6 different individuals, could be patients as well, right? Yeah, A to F. Which DNA band is the longest fragment? So we know that the longest fragments are always near the well, right? Because they are unable to move down fast. Okay, so they are stuck at the top of the gel. So band 1. Which DNA band is the shortest fragment? The shortest fragment is able to move all the way down to the gel. Which DNA bands are found to be common to all of the individuals? So which one is common to A, B, C, D, E and F? So we have 3. Okay, band number 1, band number 4 and band number 9. These are common to all the 6 individuals. Alteration of genes. Okay, we're going to look at chromosomal, structural chromosomal mutations. 5 types of mutations. Okay, P, Q, R, S and T are listed in the table. So using this list, okay, we will find out which one is P, Q, R, S and T. So multiple copies of chromosomal regions. So you have more than one copy, so these must be gene duplication or amplification. Interchange of genetic parts from non-homologous chromosomes. They are talking about non-homologous chromosomes exchanging DNA. This is translocation. Reversing the orientation of a chromosomal segment. Reversing. So for example, let's say this is your DNA. This used to be the normal DNA. Okay, there's gene A, gene B and gene C. So reversing is like that. Okay, so you have A here, but now you have C and then B. You see that they are reversed. So this is inversion. Mutation that doesn't result in any biological change. So you have a mutation, but there's no change in the amino acid. That's what this question means. No change in amino acid. So this is your silent mutation. Mutations caused by premature stop codon. So this is referring to your nonsense mutation. The diagram below shows the mutation which results in sickle cell anemia. So here you have the normal hemoglobin with glutamic acid, and the sickle cell hemoglobin replaces the glutamic acid with valine because of a mutation here, right? Supposed to be a CTT becomes a CAT. Now, so there's a change in amino acid from glutamic acid. It is now changed to valine. A change in amino acid is missense mutation. Read the diagram given below and state the type of mutations A, B, and C and the consequences of these mutations in D, E, and F respectively. So A, we would look at A, you see a mutation here, GAA is changed to GTA that results in a change in the amino acid. From glutamic acid, it is changed to valine. So this is missense mutation because there's a change in amino acid. It's a different amino acid. Let's look at the next one, B. See, TTA is changed to TAA, right? So this leads to a TAA. The mRNA is UAA. UAA is one of the stop codons. Okay, so now there's a premature stop codon. So this must be nonsense mutation. Let's look at C and F. Okay, here you see that CCC is changed to CCA, and it leads to a proline in the mutated DNA. The mutated DNA also codes for a proline. So both the normal and the mutated DNA code for proline. There is a mutation, definitely there's a mutation here. You can see that the CCC converted to a CCA. There is a mutation, but there is no change in the amino acid. So this is silent mutation, no change in the amino acid. Give an example for each of the following categories of cancer causing agents. So chemical carcinogen. Example of a chemical carcinogen will be ethidium bromide. A virus that can cause cancer can be your hepatitis B, hepatitis C, Epstein-Barr virus, can also be human papilloma virus. There are many. Radiation that can cause cancer can be your gamma radiation. This is your ionizing radiation. What are the two classes of genes which, when affected, can cause cancer? So when we say affected, we're talking about which, when mutated, okay, can cause cancer. This will be the proto-oncogenes and tumor suppressor genes. So proto-oncogenes, when they are mutated, they become oncogenes. Tumor suppressor genes, when affected or mutated, they become inactivated. A portion of DNA from two mammalian cells, one normal cell and one mutant cell, was found to have the following sequences coding for the same protein. So only the 5' to 3' DNA sequence is shown below. So this sequence is from the normal cell and this one from the mutant cell. How many base pair mutations have been introduced in the DNA sequence from the mutant cell? So we need to check. A here and there's a A here, T, G, G, there's no change in the bases so far. Okay, here, see there's a change here, right? C becomes an A here. This is okay. Let's check the rest of the sequence for any mutations. No, so only one base is changed, one base pair mutation. Identify the type of mutation introduced in the DNA from the mutant cell. So for that, if we want to know the type of mutation, we need to know the change in the amino acid. Okay, If there's a change in amino acid, it will be missense mutation. If there's a premature stop codon, it will be nonsense mutation. If there is a mutation but no change in amino acid, that will be silent mutation. So let's convert this to the RNA sequence first. Okay, RNA sequence. Okay, so the RNA sequence is going to be the same as your 5' to 3' DNA sequence, except that all the T's will be converted to U, right? U, T, T. Sorry, yeah, sorry about that. This would be A, and this would be A again. Okay, another A, U, C, G, C, A, U, A, C, G, A, U. Okay. So this is where this is the start codon AUG. 1, 2, 3 codons, and here is your mutation, right? So this is a mutation. Okay. Let's look at the what do you call it, the RNA derived from the mutant cell. Okay, so AUG as well, and GCUU, AAAU, okay, GCAU, U. Okay, so once again here, first codon, second, third, and the fourth codon is where you have the mutation, right? Yeah. So now AUC. From the normal cell, you have AUC, which is then mutated in the mutant cell to AUA, right? So AUC codes for which amino acid? Let's check the genetic code chart. Okay. AUC codes for isoleucine, ILE. AUA codes for isoleucine as well. Okay, so there is a change in the bases, so there's a mutation, but there is no change in the amino acid, so this is silent mutation. There's no change in the amino acid despite the mutation. The diagrams below show different types of chromosomal mutations. Identify the type of chromosomal mutations depicted in the diagram A, B, and C. Choose your answers from the following terms. So you have gene amplification, chromosomal inversion, chromosomal deletion, chromosomal translocation given here. So if we were to look at diagram A, what do we see? We see a chromosome 4 and a chromosome 20, and they are exchanging genetic material. So you have a part of chromosome 20 here and a part of chromosome 4 here. So exchanging of genetic material is your chromosomal translocation. Diagram B, it shows that this part of the chromosome 4 has been deleted. That is why the chromosome is shorter now. So this must be chromosomal deletion. In diagram C, it's showing us that this part of the chromosome has been duplicated. There are 2 copies now. So this must be gene amplification or gene duplication. So these are structural chromosomal mutations. Let's look at some questions from your practical sessions. Miss Jane is a research nurse doing cancer research. She wants to load a few DNA samples onto a agarose gel. She is given 3 pipettes. Oh, sorry, 4 pipettes. Given a choice of 4 pipettes, not 3, 4 pipettes. P10, P100, P200, and P1000. Write down which one must she use for each of these samples, P, Q, R, and S. Assume that each sample volume is to be drawn up only once before loading onto the gel. So if you want to measure a volume of 230 microliters, which pipette would you use? So it must be the P1000, because you can only use it once. So the P200 can only draw up a volume of maximum 200 microliters. So for 230 microliters, you need a P1000. For a volume of 3.4 microliters, we can use the P10. To measure a volume of 450 microliters, we use a P1000. And for 80 microliters, we can use either the P100 or the P200. Sorry, yeah, this should be S. Okay. A severely dehydrated patient must never be given sterile water intravenously because water is hypotonic. Okay, water is hypotonic. So what happens if you give water? Okay, you see that if you place the red blood cells here, these are red blood cells. Okay, if you place the red blood cell in a hypotonic solution, okay, what happens is that water will enter the red blood cell because of osmosis. Okay, we know that in osmosis, water moves from a region of high concentration to a region of low concentration across a permeable membrane, or semi-permeable membrane. Okay, so if you add pure water to red blood cells, the concentration of water is higher outside compared to the intracellular water. Okay, volume of the intracellular water. So water will rush into the red blood cells. The cells will swell and then rupture. If you place the red blood cells in a hypertonic solution, okay, hypertonic solution is where you have more salt on the outside and less water compared to the the volume of the concentration of water intracellular. So what will happen? Water will leave the red blood cell. Water will leave the red blood cell. So the red blood cell will shrink, okay, to an extent that it cannot do its normal function anymore. So whereas an isotonic solution, okay, isotonic solution maintains the normal shape of the red blood cell because the concentration of water inside and outside the red blood cell is the same. We have come to the end of part 2 of this revision. Good luck for your exams. Please use these slides for when you revise for your exam, but also look at your other notes, revision questions and answers, your practice questions, and all that when you revise for your exams. Thank you and all the best.